I would like to welcome Dr. Ranjan Perera. He, as Anand mentioned earlier, he is a specialist. His expertise is in genetics. He is a scientific director of genomic and bioinformatics and associate professor at the Sanford Burnham Institute. He's going to enlighten us on what we have available and the future on genetics for prostate cancer. Welcome, Ranjan.
We can extend it. Hold on a second. Where is it? Give me 1 second. Just extend this. Yeah.
Don't extend.
Duplicate. Right?
Show desktop.
Yeah, it should be duplicated.
It's okay, I can go. I can do that.
You can do it. Just cancel it. Cancel.
Well, technology. Good morning, everybody. Thank you very much for coming. I'm very humbled to be here, and it's a privilege to talk to you about what we are doing. I'm coming from one of the major biomedical institutes, as you know, Sanford Burnham Institute for Medical Research, and our mothership is in La Jolla, San Diego. And now we have our presence here in Central Florida. We started our operation in 2009. And I met during that time Dr. Patel in one of the fundraising events. That's the Jewish fundraising event of Tree of Life, actually. So we talked about, and it was very clear to me, his mission. It was clear to me that what he's thinking about what he can do additional to his patients. So how this advanced science can help him and help his disease patients and disease management. So this is why I'm here in this foundation. I'm also one of the founding members. So just to talk a little bit about the Sanford Burnham Medical Research Institute, we have several centers. As I told you, our major center is in La Jolla. We also have Santa Barbara. Now we have in Orlando. Our major strength is cancer. We are one of the NCI, National Cancer Institute, designated cancer centers in the country. So we also have a strong program going on in prostate cancer. So my responsibilities, I have triple responsibilities. I have my own research program on cancer, and I'm also oversighting the genomics and bioinformatics, which I will be talking to you in a minute, and how we can actually benefit from this type of research to prostate cancer. Now, previous speaker gave a very nice overview about the prostate cancer. There will be some overlapping of my presentations, so I think it's going to be good. So I thought of doing, what I'm going to do is, first I'm going to give you a little bit of a background which includes the risk of the prostate cancer and what are the challenges of prostate cancer and where we wanted to go, how we can overcome some of these challenges. And then we will slowly, I'll make sure that I'm not going to go into details in the genetics, and genetics is some of the subjects that my students really hate at med school. So the fight against the prostate cancer. So let's talk about the risk factors, so which I'll be talking in my next slide immediately. Then I also wanted to talk to you about how we can actually benefit from the emerging field of human genome sequencing. I think most of you have heard that human genome has been sequenced, and we know a lot about the human genome. And what is the connection of this human genome sequence into prostate cancer? And then, at the end, I will be talking about what are the key goals of Sanford, Burnham, and International Prostate Cancer Foundation. So we are looking forward to discover molecular markers for early prostate cancer detection. So this is very, very early on right now. And then also we wanted to identify markers that can help a pathologist to differentiate or discriminate indolent form of cancer versus aggressive form of cancer. So this is important because when a surgeon oncologist or the oncologist and after the surgery going into the treatment, and it's very important to understand what kind of disease management regimes he or she has to take, because what kind of therapy, what kind of drug, what is the interval, what is the dose, this is all important because we all know that one drug does not fit for all anymore, because my genome is different than your genome. And prescribing everybody like going into the doctor 500 meg of ampicillin is everybody's not anymore, right? So that is where also we wanted to push hard, go forward. So what are the risk factors? Previous speaker talked about it, and the genetic and epigenetic. So genetic is something we got from our parents, grandparents, and ancestors. There are good stuff coming in, bad stuff coming in, nothing we can do. But at least we would be able to know who we are. That's important, that's number one. Now the epigenetics is an area that the emerging science, that's very important. Epi means beyond and above genetics. So when we talk about, we know the genetics from our high school, we know the Mendel, we know the Darwins, we know the Lamarck, all talked about the genetics. The epigenetics means that the modification to your genome, think about the DNA like a strand, and there are changes going on. We talk, any changes to the DNA is a mutation, right? So we all know that. That's genetics. Epigenetics is that there is no changes in the DNA, but DNA can be modified. How you modify the DNA? You modify the DNA, I modify the DNA, not our parents modify the DNA. But it is also hereditary because of the environment, what you eat, your stress level, right? There is so much going on. So that is the epigenetics. That's the new emerging area of science to understand not only your genome, the genetics, but also the epigenetics. Then the age, we all know that. We heard about it, right? Higher, we grow older. We have high prevalence of getting the prostate cancer. Now, ethnicity is indeed a big, big issue here. Now, African Americans, as you see that, has a risk of 60% higher than Caucasians. So this is not a joke. I mean, this is exactly what Oscar is trying to do, going out and talk to people, get out and do the test, right? So this is not serious. So there is a familial component there. Then diet, of course. I mean, diet can connect it to any disease, correct? So it's not only the cancer. So what can we do, right? So there are a lot we can do. Sometimes we cannot do everything. So how do we grade prostate cancer? Prostate cancer is graded according to a score that devised and identified by Dr. Gleason. We call it Gleason Score. Prostate cancer can be graded from Gleason stage 1 score, grade 1 to grade 5. Then also there is another terminology, Gleason Score, which I'm not going into detail. The point here is this. When we do a biopsy, correct, the doctor gave the biopsy to the pathologist. Pathologist make a slice and do some magic. Basically, he's doing the immunohistochemistry. There are very standard tests out there. These are based on immunohistochemistry, looking under the microscope. So it's very important for the disease management to understand which grade is your prostate cancer, very precise grade. Because the thing is that this is extremely important for the surgeon oncologist. If your tumor is aggressive, is this the time to do the total prostatectomy or to take some adjuvant therapies? Or can we wait and see, right? So maybe you will never develop aggressive prostate cancer. So how do we do that? The standard histopathology markers sometimes may not be able to really discriminate sometimes the different stages. This is where the genetics and molecular markers, advanced human genome sequencing can come into play. So we would be able to identify using some these new technologies to clearly differentiate. The point is not to replace the currently available histopathology or therapeutic markers, but to bring some additional value for the sensitivity and the specificity. So I was the other day preparing some slides and trying to go into very details. My 13-year-old passing by, Daddy, what do you do? I said, I'm preparing some slides. To do what? I said, I'm going to talk about the prostate cancer conference. He just said that, listen, people are going to sleep. This is boring. So that gave me a point. No, I'm not going to talk about the adenine, thymine, guanine, cytosines, or pyridine, all the double-strand structures. Just the whole point here is that DNA, think about, made out of 2 strands, right? These strands have letters. That's what we talk about, genetic code or the sequence. And these letters can transcribe into another strand called RNA. And then RNA can translate into a protein. This is, I think we all know that, we learned in the high school. Now, what is happening is that this particular DNA can change, mutate. Something happened many, many years ago from your grandparents, from your parents, now you get it. So these are the red spots, these are the mutations, these are the changes. Mutations can come in various different flavors, in different parts of the gene. We call the promoters, and the coding regions, and the UTRs. I'm not going to go there. So as a result of that, here you come, right? So you come up with the cancer. So now the point is that, okay, now how do you go ahead and diagnose, right, early on? So what is the advantage of knowing that diagnosis? All these philosophical questions, we all can ask that. Okay, so we diagnose, so what is next? This is what, so diagnostic methods we talked about, there are non-invasive, digital rectal examination and PSA, prostate-specific antigen, and we all know that there are a lot of still big questions about the PSA. And invasives, that is, you take the biopsies, send to the pathologist, he makes the slides, and he uses all those, his immunohistochemistry cytokeratin markers. We call it 71720, CD57, and Ki67, all kind of things that do, and they see that, okay, it's present or absent. Now here comes that what we can bring additionally, so molecular markers. So don't worry about remembering all these things. I mean, what I'm trying to tell you that is there is another set of markers coming on the level of molecular level. SNPs stand for single nucleotide polymorphisms. That means there's a mutation, certain areas. It's very highly occurrence. You can actually group these patients into a certain population later, and then you can follow them, and then if you do the treatment, then you can say that, yes, so these are the guys who may be responding these drugs efficiently, not the other guys. CNV, copy number variation, so genes come in copies. Sometimes in cancer, you know that we call a terminology called ploidy. The gene can amplify or gene can get amplified to many. Then it's called the indel, that means insertions and deletions. So one gene from one place can jump into another place and insert and disrupt another gene, right? You're going to mess up. Deletion means suddenly get deleted and destroyed. So now you don't have that gene, right? You are going to be dysfunctional. And genes get fused. So this is a very, very, very well-known phenomena in prostate cancer. These particular two genes get fused. And then genes get mutated. These are very well-known genes in many cancers, including prostate cancers. Very recently, there's a New England Journal of Medicine, I think last month, they came up with a new gene called FOXB13. They actually see that there's a very, very prevalent mutation in this prostate cancer patient. So why DNA, RNA testing is important? The questions we can all ask, why do we need these things, correct? Number one is a relief. Okay, my prostate PSA is going so high, and doctor is telling I may have prostate cancer. So you bring another type of markers, and then you open up a little bit now, broad. Hey, what's going on? Do I really have prostate cancer? Or if I have the prostate cancer, how aggressive is that? Can I actually bring another set of markers to this currently available set of screening? Now, fever checkups and informed decisions. Now, here comes the importance of a scientist to work hand in hand with the clinicians. So until recently, what happened was these two groups are far away, right? I mean, as in nature, we are behind the screen. We are the guys who are developing the markers, developing the drugs, and the clinicians are the people you are talking to. But it's very important these two groups talk to each other. That's exactly, we're talking about the translational medicine, to translate discovery from the bench to the bedside. So this is exactly what we are trying to do with like Dr. Patel and my group, and we are trying to see what really we can do. So I can learn from him and he can learn from me. So at the end, this is the major philosophical question. What do the results really mean? So what do we do? So do we have the therapy? Do we have the cure? And what level that you can use this information? This is where we are bringing another missing link, that is a clinical geneticist or the clinical counselor. So clinical counselor is going to sit with the patient and talk about in detail about your pedigree, about where you are, your genome, and what is the importance of these changes. Now, to tell you what really we are doing, to give you a little bit of understanding, so we have the latest technologies at Sanford Burnham here in Orlando campus, as well as La Jolla, California. So we have the tools that we can go and looking into very deep. So these are standard tools for somebody who is working in the molecular biology labs. So those of you who have not been to Sanford Burnham, I invite everyone to come and visit us. So what is next generation DNA sequencing means that for the last 10 years, right, we spend $3 billion to sequence one human genome. And today, the word out there is we would be able to sequence human genome less than 1 week for $1,000. Because the technology is developing so fast, people are pushing the envelope, and there will be a time in my life, I'm sure, almost every human being will be sequenced in the planet. That's my dream. And when you go to a physician, in front of you, or him, in the laptop, the computer, your genome will be there. He would be able to see what is really going on, who you are, what's the difference. So this is basically one of our laboratories and one of the machines, just to give you an idea, and billions of reads are being generated every day. So this is a particular machine that we are using. So this is an important slide I took from Eric Green. Eric Green is the head of the National Human Genome Institute. And in fact, I talked to Eric a couple of weeks ago. We met in Marco Island. So this slide tells you a very interesting thing. So if I don't have a pointer, but I can tell you on the top panel, the red spot, you can see how the human genome started, right? And the second one is where we are now. We are trying to understand the human changes in the human genome. And the third, red spot, I think it's going up to 2020. So we will have a lot of information about our genome, the changes, the genetics, and things like that. In 2020 onwards, this is where we're going to talk about the treatment on the basis of the human genome sequencing. So we are right now in a very, very important time for us, for our children, for our grandchildren. So the science is developing so fast. So the principal goal for the International Prostate Cancer Foundation and my lab and also the institute is that we wanted to deliver a clinical diagnostic test based on genomic abnormalities that impact definitive treatment decisions. I emphasize again, impact definitive treatment decisions in men in diagnosed prostate cancer. That's the dream, and as you all know that it's not easy. I mean, those who have been in science, science is a challenge. It's not always you get the results that you want, and it costs. Science needs lots of money. So these are the things that we are trying to do. That so we are writing NCI and NIH grant proposals and Department of Defense and to raise this money to do these things. So this is the team that we are working together, high level, and this Christina Viori is the president of Sanford Burnham, and myself and Dr. Patel, and we are really, really interested in understanding and identifying in time to come the molecular markers for early prostate cancer diagnostics. Thank you very much for your attention.